Good day, everybody. My name is Leonardo Uriel González Garza. I'm a student working in the Genetics and Cell Biology department in the School of Biological Sciences of the Universidad Autónoma de Nuevo León. And we have been working on this project for the last few months, which is genetically engineering Bacillus subtilis to overexpress EPS and tapA operons to increase biofilm matrix production. First of all, we need to have an idea of what biofilms are, and basically biofilms are these complex bacterial communities in which these bacteria differentiate into different subpopulations in order to perform different tasks. So we have, for example, competent bacteria, and the ones that we will be focusing on this project in particular are the biofilm matrix producing bacteria. So biofilms in the last few years have also arose and caught the attention of the research groups, since they have new and interesting biotechnological applications, such as wastewater treatment, metabolite obtention, and even as a biomaterial in 3D printing. And in this case, there are several bacteria that have been studied lately in terms of biofilm production, yet Bacillus subtilis remains one of the model organisms to study this phenomenon. As we have mentioned, Bacillus subtilis is a biofilm producing bacteria, and it has a tightly regulated genetic network in order to control the expression of the operons related to the synthesis of the necessary machinery for biofilm production, which are the EPS and the tapA operons, which encode for the production of exopolysaccharides and extracellular proteins embedded in the matrix. So this genetic network is also tightly connected to other physiological processes in the bacteria, and sporulation is one of them. Sporulation in particular is also controlled by a master regulator, posteriorly phosphorylated, which in high concentrations, it induces entry into sporulation. So we have this problem with sporulation, since once bacteria sporulate and the spore is formed, we do not have no longer production of the biofilm matrix by that kind of subpopulation of bacteria. Thus, the development of a synthetic biology system to promote the production of a biofilm matrix and to lay entrance into sporulation is a potential strategy to maximize the obtention of this resource. And as we will observe further in this poster, this could be achieved using metabolic and genetic engineering techniques. So the objectives of this study are, first of all, to determine the potential metabolic regulators involved in the increasing biofilm production in Bacillus subtilis. Next, we have the proper design of a genetic circuit using synthetic biology techniques in order to further increase the extracellular matrix components. And finally, also use other bioinformatic tools in order to predict the behavior of our genetic circuit. So in general steps, the methodology that we will follow in this study is, first of all, conducting a research about specialized literature about Bacillus subtilis regulation of genes for matrix production and sporulation. Next, we will have the selection of the genetic parts for its use in metabolic and synthetic biology techniques to enhance biofilm matrix production. Next, as we design our genetic circuit, we would also need to design the ribosome binding site, which, if having a proper design of these pieces, we would have a proper and increased optimized functioning of the circuit. And next, we will have the construction of the genetic circuit itself for completion and prediction of its behavior through modeling. So this will be the part of the results analysis. And as we may see in the figure 1, a synthetic genetic circuit was designed under the control of an inducible promoter which is activated by the addition of high-lows into the medium. We may see that some of the main genes involved in the regulation or in this construction of the circuit are SYN-I and the APPA genes. These genes encode for products or proteins which then repress the activity of SYN-R and APRB, which are repressor proteins which are in charge of repressing the expression of the EPS and the tapA operons, which, if we may remember, are the operons in charge of synthesizing the necessary machinery for producing the biofilm matrix. Then within our circuit, we also incorporated the expository gene under the control of expository promoter. This section of our circuit would allow us to sense the increasing concentration levels of expository phosphorylated and thus reverse its process of phosphorylation, thus delaying entry into sporulation. As we may see in figure 2, the consequence that our genetic circuit would have is that once SYN I and IBBA are produced, we would then render the activity of SYN R and IBRB, thus inducing the expression of the EPS and the tapA operons. Once the genetic circuit design was finished, we also wanted to analyze how its preliminary behavior would be in a model. Therefore, we used the iBioSim tool, which helped us predict how would the circuit behave once we added hylose. In the graph, we may observe in a dark green color the addition of hylose, which we can observe as a sudden increase in the concentration and a steady line maintained along the time. And in a light blue color, we may observe the induction of the tapA operon in the genetically modified strain, while in the dark blue color, we may observe the tapA operon expression in the control strain. As we may observe, as the hylose is added, the expression of the tapA operon is slightly induced and is maintained at a higher level of production in comparison to the control strain. Finally, we may conclude that a genetic circuit was designed including the molecular regulators involved in enhancing the biofilm matrix production and delay sporulation. So we also conducted an analysis of how our circuit behaved, and we used the iBioSim tool, and we observed that there is an increase in the expression of the tapA operon, which is responsible for the production of the extracellular proteins embedded in the matrix once we added the hylose into our system. We also need to complement this study by performing further tests to assess the efficiency of the circuit in vivo in Bacillus subtilis, and if necessary, subsequent and pertinent modifications must be performed either to the experiment in general or to the circuit in specific in order to continue optimizing the production of the biofilm matrix. We also would like to thank in particular to Dr. Claudio Moreno Rocha for his support in the development of the project, to Octavio Armando Vera Tijerina for his help with the design of the different elements of the poster, and to Rodolfo Garcia Cortez for his support in the development of the model. And finally, to the FCB UANL IGEM team, of which I am part of. Thank you very much.